Hey, my name is Sethu Gunja. I'm a PhD student at Department of Cell and Molecular Biology in Uppsala University. Now I'm going to talk about the current research of our lab, telomere syndromes and biology. Telomere syndromes are a spectrum of inherited human developmental and degenerative disorder with telomere dysfunction. This includes idiopathic pulmonary fibrosis, which is an adult lung disorder. Aplastic anemia is a bone marrow failure syndrome. Dyskeratosis congenita (DC) is a severe form of aplastic anemia. Hural Henderson syndrome (HHS) is a severe form of dyskeratosis congenita. Both DC and HHS patients are diagnosed with its classic symptoms like nail dystrophy, abnormal skin pigmentation, oral leukopenia, and severe bone marrow failure. As I said, these disorders results in telomere dysfunction. The onset and severity of the clinical symptoms of these disorders correlates with the telomere length. That means, shorter the telomeres are, more severe and early onset of the disease. These disorders are mainly caused by the mutations in the genes that encodes the components of the telomerase enzyme or telomeres. For example, TERT, which is a reverse transcriptase in telomere enzyme complex, and other telomere binding proteins. So, mutations in any of these genes can cause all these disorders depending on the onset of the clinical symptoms. Recently, we have found a patient that is suffering with hural Henderson syndrome that has mutations in a gene called PARN. So, what is PARN and how does it relate to telomeres? PARN is one of the major mRNA deadenylation enzymes. Biochemically, it is well characterized, but the physiological relevance is not clear. The unique property of this enzyme is it is present in both nucleus and cytoplasm. In cytoplasm, PARN is found to be involved in the removal of poly A tail by binding with the both 5' cap and 3' poly A tail of the mRNAs. Whereas in the nucleus, it is involved in the maturation of the small RNAs, for example snoRNAs, by trimming their 3' ends. TERC RNA is an RNA molecule in the telomerase enzyme complex that acts as a template for the synthesis of the telomeres. It is also one of the type of snoRNA. Due to the defective PARN activity in our patient, the mature levels of TERC RNA were dropped and the immature levels were increased significantly. And due to this defective TERC RNA maturation, these telomeres were extremely short in our patient. So, to further study the effects of PARN deficiency throughout the lifespan of an organism, we need to develop a stable model system. For that, we choose CRISPR-Cas9 technology, a powerful genome editing tool, to introduce the mutations of our patient in the popular model organism zebrafish. Using these zebrafish model systems, we are able to study the physiological role of PARN, and we can also follow the development of the disease state over several generations. Thank you.